Hi, this is the Biology Paper 2, or Paper 1 rather, Foundation paper for 2022. Question number 1. Foods are digested before they are absorbed into the blood. Okay, so they're digested to make them smaller. Okay, because if they're too big, they can't get absorbed into the blood. Figure 1 shows the organs of the human digestive system, right? So before I can even think about it, so we've got the mouth. That big thing there, I might even colour that kind of darkish red, is the liver. Then behind the liver we've got the stomach, and then that kind of spaghetti looking thing is then the small intestine. So which organ is the stomach? Maybe C, tick C. What type of enzyme is produced in the stomach? Right now, I could go through all the kind of enzymes, and you do need to kind of know what enzymes are produced in each one. So for example, the mouth has got a carbohydrate in it. When you get down into the stomach only has one type of enzyme in it, and that enzyme is pepsin, right? And pepsin is a protease, because pepsin breaks down proteins, and they're unusual because they work at pH 2. Right, the next question. What term describes the pH of stomach, right? Of the stomach, right? So 7, 1, 14. 7 is neutral. 14 is alkali, and 1 is acidic. Okay, and obviously you've got red at this end. Okay, you've got green in the middle, and you've got purple kind of at that end there. Give one reason why the stomach is this pH. So what term describes it first? It is acidic. Okay. Give one reason why the stomach is this pH. The main job of the stomach, right, is, funny enough, it is to sterilize food. Right, so when your food goes in from the canteen, what it does, it goes down into your stomach, hits the acid, and it kills all the bacteria on there. Okay, that's then the main reason why stomach acid is that pH. Next question. Which organ produces bile? Right, so bile is definitely not the large intestine, because that job is to absorb water. It is definitely not the mouth, because that would be just grim. It is not the pancreas, because the pancreas only produces enzymes. So it is the liver. And what it does, the liver produces bile, and then it stores it in the gallbladder. Okay, and then when it's needed, it then comes out of the gallbladder. How does bile help digest foods? Now, I always teach it as emulsifies fats. Okay, it's a bit like when you have a bacon butty at night, or something that's very oily food, and what it does, it goes straight in the sink, and you've got kind of oil globules all kind of over the top of the water. Chuck in some fairy liquid, and what the fairy liquid does is it breaks it up, it emulsifies it. So what happens with this is, how does bile help digest food? Increases the surface area of fats. It is that. So it is an enzyme, it's definitely not an enzyme, and there's a big clue with enzymes, because enzymes usually end with ASE. It makes the pH of the small intestine acidic. No, because what it also does, it also neutralizes the acid from the stomach to make it neutral. A student tested different foods for the presence of protein, starch, and sugar. Right, protein is Biuret. Okay, and what it does, it goes blue to lovely lilac-y kind of colour, a bit like this kind of colour, except maybe a paler version of it. Starch test is iodine, and that goes from kind of an orangey colour, orangey brown colour, put brown there, to blue-black. I don't know why it says blue-black, it just looks like black to me, right? But if the syllabus says blue-black, you write blue-black. And sugar is the Benedict's test, right? And I like the Benedict's test because it goes blue, but then I can never remember the order, so I'm going to put red over here. I know it starts at blue and then it goes red, and then it goes orange, yellow, green. red, orange, yellow, green, blue, colours of the rainbow backwards. All right, anyway, that's sort of beside the point. Draw one line from each food, I'm going to do this in red, molecules to the reagent used to test for the food molecule. Right, now what I'm going to do is I'm going to do it right, but I'm going to do it wrong. Right, you'll understand why, so just don't do this. So protein, I know from over here it's the Biuret, and we're going to go, whee, yep, that's correct. Starch, I'll do it with a different colour, because that's great. Starch, I'm going to go all the way up there, around there, don't overlap at all. Iodine. And sugar. Sugar, go all around there, around the houses, right the way around to there. So what I've done there is I've done it right, but you never ever do it like that. What you do is you get a ruler. Now obviously I haven't got a ruler here. Okay, so you get a ruler and you just draw a straight line there. That's a perfect straight line, as you can tell. Starch, that is also a perfect straight line. And sugar is also a perfect straight line, because what the examiner does, they look very, very quickly at it. And if there's loads of wiggly lines and stuff like that, they should still look at it, but you just kind of never know. Give one safety procedure when using Benedict's solution. Okay, one safety procedure. So what I'm going to do is I'm going to chuck in maybe 3. Okay, Benedict's solution. Now, I don't really know much about it, right? All it does is kind of tests for sugar. All right, so goggles. Okay, I'm sure if I get it in my eyes, it's never going to do me actually any good. You need to wash it up. Don't drink. Okay, well, it's probably not the best one to do, that one, because it's not really a safety precaution, right? So it's not sort of something to do with a practical. So maybe don't put that one in. But goggles, wash up, and also make sure you use a water bath in the actual experiment itself. 1.8. Table 1 shows the results for one food sample. Okay, so colour after the test, red, blue, black. Okay, now that's worked. Biuret's was blue to start with, and Benedict's red means there's lots of sugar. Which of the tests showed positive results? Okay, I've just answered that. All 3 tests, well, no, because the Biuret didn't show a positive result. Benedict's and Biuret only? No, because Biuret didn't. Biuret's in that one as well. Benedict's and iodine only? Correct. Again, I've only ticked one box. Starch molecules are not absorbed into the blood from the digestive system. One reason why. Okay, I'm going to draw starch. Here's starch. I could keep on going forever and ever. Right, there's starch. Starch is big. And the whole point in the digestive process is to break it, break it, break it, break it, break it. So then they're small enough to be absorbed. So starch molecules are not absorbed into the blood from the digestive system. Give one reason why. Starch molecules are too large to be absorbed. Okay, fabulous. Question number 2 shows a section through a leaf. Okay, let's do it in green because obviously it's a leaf. So on the top there, we've got a waxy layer, and what that does, it prevents the loss of water. Okay, it doesn't prevent water getting in, even though it does a little bit. It prevents the loss of water. Again, they've got the top layer there, the epithelial layer, right, and that is then for protection. Palisade layer, which is obviously important in this question. We've then got air gaps. That is then the spongy mesophyll. Mesophyll. I hope that's spelled right. Is it double L? Not sure. And I often say it's spongy, right, because it sort of says spongy. Then at the bottom there, we've got X. X is pointing to the little hole, which is the stomata. And then on either side of it, right, with the little eyes kind of staring at you, are the guard cells. Right, let's go to the questions. Give one way the palisade cells are adapted for photosynthesis. Right, they have got lots of chloroplasts. Okay, They are also at the top of the leaf. And they also are kind of rectangular shape, rectangular, to allow many to be packed in. Okay, but the best one on there is lots of chloroplasts. They're at the top of the leaf, right? That makes sense, doesn't it? It makes sense to have all the chloroplasts at the top of the leaf. Next one. Let's change colour now, border green. Gases pass into and out of the leaf through small pores in the surface of the leaf. What are the small pores called? Well, they're not guard cells, because a pore strikes me as being a hole of some sort. Guard cells are on either side. Xylem is definitely not a pore, so it is stomata. Stomata are the little holes. A student viewed a section of leaf using a microscope. Okay, I'm thinking investigation here. Okay, I'm thinking required practical. So what you've got to do now is you've got to think about that required practical. Right, you've got to think about that required practical that you then did doing microscopes way, way, way back. The student measured the length of one of the palisade cells. Fantastic. The cell image measured 28 millimeters. Right, so image, best thing to do now is pick out the data, 28 millimeters. And viewed it with a magnification of times 400. Fabulous. Okay. Calculate the real, so we need to work out the real length of the palisade cell in millimeters. Now they're being nice to us there, right, because what they're doing is we've got that in millimeters, so they want this in millimeters. So here's the equation. Let's substitute in. I'm going to go real equals image is 28 divided by the magnification is 400. Okay, and what that works out to be, it equals 0.07. And because that's in millimeters now, the real length will also be in millimeters, 0.07 millimeters. So literally all I've done is I've substituted in and then done the sum. Next part, convert. Oh, here we go, convert. The real length of the cell from millimeters to micrometers. Oh, they even tell us that. So 1 millimeter equals 1,000 micrometers. So if I've got 0.07 millimeters, think about it now. If 1 millimeter equals 1,000, is this number going to get bigger or smaller? Right? Well, without any shadow of a doubt, what it's going to do, it's going to get bigger. So what you do is you do 0.07 times by 1,000, which gives me 70 micrometers. Carbon dioxide can move into and out of cells. What is the process by which carbon dioxide can move into and out of cells? Right, oh, they've got all three there, look. All three. Active transport needs energy, and it definitely isn't that one. Osmosis, that's water, so it must be diffusion. So diffusion is the movement of particles from a high concentration to a low concentration, down a concentration gradient, and doesn't use energy. And that is what happens in a leaf. Ooh, lots of dots. So what we've got here is shows a diagram of 4 cells, A, B, C, D. All right, now I can see that there's different amounts of dots around each one. Each cell is surrounded by carbon dioxide molecules. So I assume that the dots inside are carbon dioxide and the dots on the outside are carbon dioxide. Yes, that's what it tells me here. Which cell will carbon dioxide move into at the fastest rate? Move into, right? So I've got my cell there like that. It's which one carbon dioxide will move into the fastest. So diffusion is the movement of particles from a high concentration, so I want high outside, to a low concentration. Okay, so if I now look at this, so what I'm doing here is I'm looking at this one, they're the same, so realistically diffusion is not going to happen. D, there's more inside, so they're going to move out. C, looks like to me the same, so there's going to be no movement, no net movement. A. And that is because there is a steeper diffusion gradient. So you think about your definition that you keep getting asked about all the time. And there is, I'll just write it here, there is a bigger difference in CO2 outside to inside. Now really I've done the same thing twice there, okay? But obviously a steeper diffusion gradient is a good answer because it's very scientific, but it also tells you the correct answer. Oh, here we go, investigate. Okay, I love it when it says investigate because I know full well I am going to be talking about a required practical. A student investigated the effect of different colours of light. Oh, different colours of light. We did different distances. So they're trying to catch things out a little bit here. So we're talking about colour of light on the rate of photosynthesis. Figure 4 shows some apparatus the student used. Now I'm familiar with that. I've seen that before. That's pondweed at the bottom there. All right, that's a boiling tube. So there's pondweed. So do it green. It'll be nice and green. We've got a boiling tube going over the top here. There's a funnel, and then there's bubbles of gas that never appear in the real thing, but they're obviously on the diagram. And in there we've probably got water, and also in there we've probably got sodium hydrogen carbonate. Okay, which is the substance that sort of provides carbon dioxide. And then they've got a little picture of a timer. How interesting. The student placed the apparatus in blue light. Why did I do that in red? Blue light. Oh, then green light, and then red light. The student measured the rate of photosynthesis in each light colour. What two measurements should the student make to calculate the rate? Now, rate always needs a time, okay, because it's always meters per second, bubbles per second, sausages per second. Rate always has a time at the bottom. So therefore, my first measurement is going to be time. And the second measurement, it's labelled for me, is bubbles of gas. Oh, gas. Or we could have it as volume of oxygen produced. Okay, because what we're doing is we're collecting it there rather than just counting the bubbles. Question 2.7. Okay, give two variables the student should keep the same in the investigation. Control. Okay, so there's a number of different ones you can put in here. It says 2 on there. I'm going to put a few more. Temperature. Okay, temperature has to keep the same. That's why in the actual practical itself, you always have to put like a screen in between or use an LED light. Another one is light intensity. Okay, so don't just put the light, you put light intensity. Distance to the pondweed. Okay, and the concentration, oh, this is one I mentioned before, of sodium hydrogen carbonate. Fabulous. These questions are the best, right? Because what they've done is they give you a piece of graph paper, but then they say it's 4 marks, and then what they do is they tell you what each mark's for. So all you've got to do is you just got to go down that list there, and if you go down that list and do each one, then you get 4 marks. Right, complete figure 5. Here's figure 5. Label the y-axis. Okay, so this is the y-axis. If I look at my table that I've then put over here, I can see that colour of light is at the bottom, so therefore I am going to write the full thing, rate of photosynthesis in arbitrary units. Okay, and you're completely and utterly daft if you don't write the whole thing, because that's obviously what they're asking you to do. Tick, just got 1 mark. Fabulous. I know now I've got 1 mark in the whole paper. Use a suitable scale. All right, so I'm going to start at 0, and then I'm going to go up to 9 is my maximum. So let's think about it. So I could go 1, 2, 3, 4, 5. Well, that's obviously stupid. I could go 2, 4, 6, 8, 10. That fits. So I'm going to go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There we go. Tick. Suitable scale because it goes up nice and easy. It's all even. Okay, there's no, I'm going to do number 1, then 8, then 9. Right. And it goes right the way to the top. Suitable scale. 2 marks. Fabulous. Right. Plot the data from table 2 as a bar chart. Okay, so what I'm going to do is I'm going to do blue. Why did I do it in orange? I'm going to do green. And I'm going to do red. Okay, so I'm going to start there. 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 So then what I've got is it's nice and evenly spread. There's an even gap at the start. There's an even gap in between each bar. So blue. Now obviously mine's going to be a little bit messier because it's difficult to actually use a ruler on this, right? So what I'm going to do is I'm going to go up to 9, I'm going to go across there. See, I've messed it up already. Okay, and I've got a wiggly line going all the way down, right? You're obviously not going to do a wiggly line, you're going to use a ruler. There's my blue bar. Then I'm going to go to green, and green is 1, so I'm going to go 1, and then red is then 8, up to 8, and then down again. Plot the table. Fabulous. Label each bar. Okay, I did that already. Got blue, green, red. 4 marks, and I know I've got them. Look at table 2. What colour of light should be used to grow plants in a greenhouse? Right, well, green did almost no photosynthesis, red did 8, blue did 9, so therefore the best one is blue. This question is about disease, okay, nothing better than talking about a disease. Rose black spot, I wonder what plant that grows on, I wonder what it looks like. Right, is a disease where black spots, should we start doing in black, where black spots develop on the leaves of a rose plant. Okay, I just sort of said that. What type of pathogen causes rose black spot disease? Right, this is one you just got to kind of remember really, right? So you need examples of each one, and it is a fungus. Plants with rose black spot disease often have yellow leaves. Okay, so they've got yellow leaves. Suggest one reason why the leaves are yellow instead of green. Yellow instead of green. Okay, they're normally green because of chlorophyll. Okay, so that's the substance then that causes the greenness. Okay, so if they're yellow, they have less or no chlorophyll. And that's kind of the reason for it. They're yellow because they've got less chlorophyll. Next one, explain why plants with yellow leaves grow slowly. Okay, if they've got yellow leaves, they're going to grow slowly. They've got less chlorophyll, so therefore there is less photosynthesis. Right, what you've got to do is you just got to use the right words as well. Okay, I'm using the word photosynthesis there. You could say less food is made, and you just wouldn't get anything for it. So just use the right words. Also, less light is absorbed, right? Because what does photosynthesis need? It needs light. So therefore, okay, see what I'm doing here? I'm doing my explain. I've given a reason. And now that I'm doing a therefore, or I'm doing a so, or I'm doing a because that's what an explain question asks for. Therefore, there is less glucose made because there's less photosynthesis. And that is then because there's less photosynthesis as well. But the key to that one then is you've got to see I kind of split it into 2. When you get an explain question, you kind of do a describe first, which is this is the reason why. And then I always like to then start my explain questions with so, because, or therefore. So what I could do is I could say, There is less photosynthesis, therefore less glucose made, 2 marks. There is less photosynthesis, so there is less glucose made, because doesn't necessarily fit into that one. Okay, but that's kind of a crux behind it. 3.4, get your ruler out. Okay, the spread of Rose-Black's blot can be controlled by using different methods. Draw one line from each method of control to the explanation of how it works. So that means that when you've got your boxes there like that, okay, now I have seen this so many times before, oh, I don't quite know the answer. Okay, so what you've done 2 lines from each one of them, right, you get a big fat 0, okay? Right, so remove and burn infected leaves. Remove and burn infected leaves. Creates a barrier. No. Pathogens are killed. Yeah. Okay, burning tends to kill pathogens. Just check the others. Remove and burn infected leaves. Reduce the chance of pathogen by water droplets. No. Reduces the... No, obviously not going to reduce the temperature. Second one. Water the roots of the plant only, not the leaves. Okay, that makes sense, doesn't it? If you're going to water the leaves, then the fungus is then going to spread around. So water the leaves, the roots of the plant, not the leaves. Barrier, no. Reduces the chance of pathogens being... Well, it tells you it in there, doesn't it? Reduces the temperature, no. Tobacco plants may become infected with a pathogen called TMV. I honestly cannot remember what TMV stands for. Okay. What type of pathogen is TMV? Oh my word. So TMV. Right, so it's a tobacco plant. Well, tobacco, I don't know, tobacco V. Tobacco, I think it is mosaic. Bacteria? Obviously not. Fungus? Protist? Virus. Tobacco mosaic virus. So it must be a virus. Malaria is caused by a protist. How is the malaria pathogen transferred to humans? Mosquito bite. Okay, horrible, nasty things. How can the spread of malaria pathogens be reduced? Tick 1 box. Malaria. Okay, so you've got to remember malaria is by an insect bite. Okay, and it kind of destroys your liver and destroys your red blood cells, and then it gets bitten again and another mosquito takes it in, and it's like a horrible, nasty cycle. Sexual contact. Well, malaria isn't a sexually transmitted disease. Malaria, they get bitten by a mosquito, therefore it's nothing to do with cooking food. Drain water from swamps. Use a tissue when sneezing. Without any shadow of a doubt, it's drain water from swamps, because that's where malaria mosquitoes live. Cigarette smoking, disgusting habit. Right, is the main cause of cancer in the UK, right, and people still do it. Mutations in cells cause cancer. Where in a cell do mutations happen? So there's my cell, there's my nucleus, I've got cytoplasm, I've got a cell membrane around the outside, I've got ribosomes in there, okay, I've got mitochondria in there, and there's my nucleus. Okay, where do they happen? Well, they definitely don't happen in the cell membrane, because that's just like it's almost like a bit like just a layer around the outside. It doesn't happen in the cytoplasm, because that's where chemical reactions happen. Nucleus, DNA is where mutations happen. Why do some cancers develop into large tumours? Okay. Now, the thing with tumours, cancers, is you've got benign and you've got malignant. Okay, benign are the ones that are reproducing rapidly, but they're kind of contained. Malignant are the ones that are reproducing rapidly, but they kind of have little roots that kind of come out of them, and they can kind of spread and they can go into different organs. Large tumours. Cells never stop dividing. Okay, yeah, I kind of just said that. Cell respiration is slowed down. Well, if respiration was slowed down, it wouldn't grow as fast. Enzyme activity is stopped. Tick one box. Cells never stop dividing. Cigarette smoking has been linked to many different types of cancer. Oh, right, so you don't just get lung cancer, you get liver cancer and blood cancer and all sorts of different cancers. It's a smart thing to do. Lung cancer is the most common type of cancer caused by smoking. Suggest one reason why. Okay, I need to do this in red really, don't I? Okay, so the main reason why is the chemicals from the smoke are in direct contact. Okay, so they're in direct contact and they are more likely to cause the damage to the DNA. A person with lung cancer can develop secondary cancers in other parts of the body. Describe how this happens. Right, so cancer cells can be transported in the blood to find other places to embed. Right. So what you have is you have a benign cancer that's like that, and it just kind of grows and gets bigger. You have a malignant cancer, and it kind of has little roots that are coming off. Now, it's not quite as obvious as that, but what can happen is one of those little bits of root there breaks off, goes off in the blood, finds the liver, embeds in the liver, and then what you do is then get a secondary cancer, and that's when you're in a lot of trouble. Sometimes a person may need a lung transplant. The National Health Service, right, will not offer a lung transplant to a person who smokes. Sounds harsh. One reason why. Okay. Smoking will damage a new lung. Okay, so you're a smoker, you don't stop, you get a lung transplant, you get a new lung from somebody that's never smoked, you carry on smoking, you just destroy the new lung. Why should you have it? There could be someone who needs a lung that doesn't smoke. Okay, this sounds very kind of harsh and stuff like that, but you've got two choices. One person doesn't smoke, one person does smoke. Who's the best person to give it to? Okay, and the NHS think that lung cancer, if you're a smoking illness, is self-inflicted. What graph? I like graphs. Graphs are cool, right? Because you can look at a graph, you can get loads of marks. Figure 6 shows data about skin cancer. So we're not talking about lung cancer, and we're talking about skin cancer in males. For different age groups in the UK, the data shows the number of new cases of skin cancer in a year. So we're talking about skin cancer in men and new cases. This tells us down the side here, number of new cases of skin cancer, 0 to 1,200. Age in years, and it goes from 15 all the way up to 90 plus. Okay, and I can see that one's big, that one's small. I can see there's a pattern that kind of goes up there like that, and I can see there's a pattern that goes down there like that. How many more new cases of skin cancer are there in males aged 40 to 44 than males aged 15 to 19? So let's look at the data. 40 to 44. So if we look at 40 to 44, the number is 320. Now I'm not going to put it on there, but what you do is you get a ruler and you go right the way across and you work out exactly what it is there, and you do it as precisely as you possibly can. You might just go 5 out and you might lose marks for it. And then males age 15 to 19. So the smallest one, okay, I can then see that that is 20. How many more new cases, right? So make sure you're writing your sum on the lines. So it would be 320, take away 20 equals 300. So the number of new cases, more is 300 new cases. There are no new cases of skin cancer diagnosed in males younger than 15. Fabulous. Suggest one reason why. Under 15, there has been little exposure to ionizing radiation. Okay. Probably a more likely one to remember is little exposure to UV light. Also, mummy and daddy will get you when you're younger to put on more use of sunscreen. And because it actually takes quite a long time for kind of cancers and things like that to develop, there has been little DNA damage that's happened and not enough time for damage to show. Okay. Ooh, back to the graph. Well, we've talked about this graph already, can't bother to talk about it again. Give one conclusion from the data in figure 6. Okay, so that's figure 6. So what I drew on before is I drew that. I drew that. I highlighted that. So let's just think about it. Most new cases age, let's just check it out, 65 to 69. Okay, that's a fact. Conclusion, one conclusion. The rate increases, sorry about my writing, from 15 to 19 up to 65 to 69. Okay, I can see that. Rate decreases after 65 to 69. So the trick to a question like this is, if they ask for a conclusion, is you look for any patterns. You look for a pattern, you look for the biggest, look for the smallest, look for any kind of going ups, going downs, right? And then what you do is then you sort of add numbers in to give you a conclusion. Survival rates of all types of cancers improved over the last 20 years. Brilliant. Okay, 2 reasons why. Treatment is better. Okay, there's all sorts of different radiotherapies and chemotherapies that are being used. The drugs have improved. We can get earlier diagnosis. Therefore, or in addition to that, is there's better screening. Okay, so women of a certain age get letters through the post saying breast cancer screening. Okay, and they might not even suspect it, but if they're doing screening en masse like that, then they're going to catch them. And also, better doctor knowledge. Okay, better doctor knowledge, which means doctors can spot it better, right, and they've got better knowledge about how to treat it. Now, the following questions, right, what I've done is I've just adjusted the numbers on them, right, using the pen, because these are then the crossover questions. These are then the questions that are going to make the difference between whether you're getting 2s, 3s, to whether you're getting 4s or 5s on the foundation paper, right? So these 2 questions that follow on from here are the first 2 questions then on the higher paper, right? But what you've got to do is you just got to make sure you follow exactly the same patterns I've talked about before, and you put your knowledge onto your piece of paper. Now, question number 1. Now, what I've done through this video is I've just split it up into every single question, all right? And then what I'll do is I'll then talk about exactly what my train of thought is.
So question 1, right, bacteria can cause a variety of different diseases in humans, right? So my first thought is I'm then going to draw a bacteria, right? Because of what I reckon is, right, I reckon that there's going to be something to do with the structure of it. So that's the cell membrane, no, cell wall, God, stupid, cell wall, that's the cell membrane, they are then flagella going around the outside. Now that looks like a nucleus the way I've drawn it, but that's a plasmid. That is then sort of DNA material that's in there, and then it's got cytoplasm, because a bacteria is a prokaryote, so it has no nucleus. Now let's read the question. What are two similarities between a bacteria and an animal? Right, animal cell. Right, there's an animal cell, so there's the nucleus, there's the cytoplasm, it's got ribosomes, it's got mitochondria, and it's got a cell membrane. Okay, two similarities between a bacterial cell and an animal cell. Tick 2 boxes. Complete and utter plonker if you don't tick 2 boxes. So both have a cell membrane. Yes, yes, tick. Both have a cell wall. No, an animal cell doesn't have a cell wall. Both have a nucleus. Bacteria don't have a nucleus because they're prokaryote. Both have a cytoplasm or cytoplasm. Yes, yes, tick. Just to check on the last one, both have plasmids. No, animal cell doesn't have plasmids. Question number 2, salmonella food poisoning is caused by bacteria in food. Okay, so we're talking about bacteria again, right? We're talking about salmonella, nasty food poisoning. Okay, I've had salmonella food poisoning a few times, not particularly very nice. Give one symptom of salmonella food poisoning. All right, so I'm just going to think about it now. I remember now I had vomiting and I had diarrhea. Okay, I've put 2 symptoms down, but they're both valid. Oh, do not refer to vomiting or diarrhea on your answer. Good job I read the question. Scribble them out. Okay, so fever, stomach cramps, sweats, chills. Okay, all those different ones could then be put down, right, as one symptom of salmonella. And what you do is you put the one that you think is most likely going to be the one that is correct first. 1.3, what is the name of the first antibiotic developed? Right, there's no logic to it, right? It is penicillin, right? That Fleming guy, I think, right? So what we've got is we've got penicillin, and that's one that you've just got to remember. Okay, it's just like a fact. Oh, a graph, cool. All right, so let's have a look at it. Right, before we even think about any questions at all, let's have a think. A child with severe bacterial, right, and antibiotics. Right, now we know that bacteria are affected by antibiotics, but viruses, protists, fungi aren't affected by antibiotics. Figure 1 shows how the concentration of live bacteria in the child's body changed when taking a course of antibiotics. Right, so we know that if you take antibiotics, it's going to get rid of the bacteria. So let's have a look at the graph. Time in days, so it goes up to what, 4.5 days. That is when the antibiotic was then started. Concentration of live bacteria in the body in arbitrary units. So arbitrary units kind of means just like a made-up unit, it's just kind of relative. And what it starts at is it starts there, which is about 3.2 arbitrary units. Now I'm looking at that. If I start the antibiotics, I reckon there's going to be something to do with the fact that it goes up. Why would it go up if you've just started taking your antibiotics? Don't get that. Okay. The concentration of live bacteria in the body continued to increase after starting the course of antibiotics. Right. So what it is actually doing there, it is actually asking me about that. Suggest one reason why. Live bacteria are still reproducing. Right, so if you think about it, at this point here, right, the bacteria are still reproducing. So it's going to take a little bit of time, right? In fact, it takes almost a day for the actual antibiotics to start killing more bacteria than are actually being reproduced. So you could put down there is a time delay, right? Because there's a time delay before you have it. All right, and they are then the two kind of main reasons, and it sort of makes logical sense. You take the antibiotic, it takes a little bit of time before it then starts. So if you had a headache, and what you did is you then took paracetamol, your headache could get a little bit worse because it's kind of getting worse, and then it then goes down. 1.5, after 3 days of taking the antibiotic, the child felt better. Fantastic. There were still bacteria in the child's body. Why did the child feel better? Well, if there's still bacteria in there, right, then there is kind of going to be a thing. So now if I think about bacteria, right, how do bacteria, before I read any question here, how do bacteria actually make me feel ill? What they do is they produce toxins, right? And it's those toxins that are actually making me feel poorly. So let's have a look. Why did the child feel better? Tick one box, right? If you tick 2 boxes, you're a plonker, right? If you tick no boxes, you're also a plonker, because if you've got no idea, you might as well guess. Bacteria had become immune to the antibiotic. It is never, ever, ever, ever going to happen that quick. The child had become resistant to the bacteria. No, right? That's again not really possible for that to actually happen. There were fewer toxins in the body than in day 0. Correct. Question 1.6. Suggest why doctors do not give antibiotics to patients with minor infections. Right? If you've got a minor infection, right, what's probably going to happen is you're probably going to get better anyway. So there's almost no point in actually even going to the doctors in the first place because you are going to get better. Also, a minor infection could be a virus, right? And if it is a virus, antibiotics aren't going to do anything anyway. So let's have a think about it. Suggest why doctors do not give antibiotics to patients with minor infections. Now, what we're going to do is we are going to prevent, so we don't give them to prevent resistance to antibiotics. So we don't give antibiotics. You only give antibiotics when you really need to, right? Because the more times you give it, the more chance that the bacteria are then going to become resistant to it. And if they become resistant to it, life gets harder. It's actually in the answer sheet here is you're probably going to get better anyway. And it also could be a virus. So then 3 possible answers that you could have. Right? Don't give antibiotics because number 1 is you don't want the bacteria to become resistant. You could get better anyway, and it could be a virus. Right, figure 2 shows blood viewed using a microscope. Right, blood. Right, let's think about blood. I'm going to do it in red. Right, I'm going to do what's in blood? Red blood cells. Okay, what are they? There's lots of them. Right, they're donut shaped, so they're like a disc. Right, predominantly more than most. What else have we got in it? We've got platelets. Okay, so what they are is they are fragments. So if they are a fragment, that probably tends to mean that they are small, and their job is to then clot blood. Job of the red blood cell, carry oxygen. What else have we got in here? We've got white blood cells. Okay, we're doing white, but it's got a white background. White blood cells, they tend to be bigger and not many, because most of the time you don't really need very many. And the final bit is plasma. Plasma is kind of like a straw-coloured liquid, right? And what it is, it's all the liquid kind of in between these different things. So if I look at these now, A is probably a red blood cell. B, bigger and not as many, white blood cell. C is the liquid in between, plasma. D, small little fragments, platelets. right? So before I've even gone through the question, what I've then done is I've gone through my knowledge, right, of what is in blood. Right, a vaccine will stimulate the production of antibodies. Which part of blood in figure 2 produces antibodies? Well, it's not red blood cells, it's not plasma, it's not platelets. White blood cells are the sort of immune system. All right, so tick 1, B. Which part of the blood in figure 2 starts the clotting process? Okay, I know that already. Small fragments, D. Question number 2. This question is about cell division. Right, before I read anything else now, what I'm going to do is cell division. So cell division as a center, right, pie chart. That then there is interphase. That there is mitosis. That there is cytokinesis. Can't fit it in very well. So interphase is kind of where all the mitochondria, the ribosomes, the amount of DNA that's in a cell doubles, and it goes all the way around there. Mitosis is then where the actual DNA itself splits, right? There's that full process, isn't there, of what happens in mitosis. And cytoplasm is then when the whole thing splits into 2, and we get 2 different cells at the end. So let's have a look at the question. The question is about cell division. Okay, I've just done it over there. That's cell division. Write the biological structures from the box in the correct order of size. Oh, it's got nothing to do with that. Okay, so we've got cell, chromosome, gene, nucleus. So it's something to do with a cell. So what I'm going to do is I'm going to draw a cell, and I'm going to draw the nucleus in it. And then what I'm going to do is I'm going to look at the question, and it says there smallest, and it says there largest. Now, my logical brain says biggest first. Okay, so what I'm going to do is I'm going to start big. So which is the biggest one out of all these? I've just drawn it, a cell. Inside the cell, I've then got that there, the nucleus. Inside the nucleus, so if I then zoom in on the nucleus like that, what I've got is I've got those little strandy things. And what those strandy things are chromosomes. Chromosomes. If I then zoom in on a chromosome, each one of these little tiny bits on there is a gene. There you go, smallest to largest. Figure 3 shows how a fertilized egg cell can produce specialized cells. So specialized, right? So what we have is an animal cell, and a specialized cell is where they change to be able to do a specific role. Okay, they sort of they become different, right? So what they do is they become different to do a role. Right, so fertilized egg cell. Fertilized means we've had an egg, I don't know why I did that, and a sperm. They've then come together and we've got a fertilized egg cell. Then what it does, it starts to replicate. That is then when it becomes an embryo. Process A, now I'm pretty confident that what they're going to do is they're going to ask me what process A is. It could become a red blood cell, it could become a muscle cell, it could be a nerve cell, right? So they're the 3 specialisms. Oh, surprise, surprise. Name process A. That is differentiation. Okay, so that is then the actual process that is then expected, is differentiation. How many cell divisions are needed to form a 16-cell embryo from the original fertilized egg? Right, there's a fertilized egg. And then what they do is they split. So when they split, I then get 2. So that is 1 cell division. Then what they do is they split again. So that's the second cell division. How many am I after? 16. Then they split again. So 4 then becomes 8, which is 3. 8 then becomes 16. 4 cell divisions. 2.4. In humans, a fertilized egg contains 23 pairs. Pairs means 2, so therefore if there's 23 pairs, there must be 46 chromosomes. All right? Fertilized egg, 23 from the egg, 23 from the sperm. Right? That's my thinking. How many chromosomes will there be in each embryo cell? So in one embryo cell, there's 23 pairs. That means there must be 46. Here we go. Here we go, cell cycle. Okay, I've just drawn it then. Stage 1, stage 2, well, they've named it mitosis in that one, stage 3. And that there is the start. Okay, so I drew it before, didn't I? So stage 1 is interphase, where all the ribosomes and mitochondria and the amount of DNA doubles. Mitosis is then where the nucleus kind of doubles, the DNA in it. And stage 3 is cytokinesis. Describe one change in the cell that occurs during each of the stages of the cell cycle. All right, so what do we think is going to happen in that? It's called interphase. Right, now that sounds posh and that sounds like I'm being really, really clever, right? But that isn't a change. All that is the name of it. So it's a bit irrelevant, really. So what actually happens in there is we have the subcellular parts increase, right? So your ribosomes, your mitochondria, your cytoplasm, all those kind of things increase. And what you can do is you can also put in there ribosomes increase, mitochondria increase, DNA increase. So what I've done then is I've done the name of it, where I actually don't get any marks for at all. So what I'm going to do is I'm actually going to scrub that out because that's rubbish. Subcellular parts increase, that's just like posh saying that ribosomes, mitochondria and the DNA increase. So that's stage 1. Stage 2. Give one change in the cell that occurs during each of these stages. So what I've now got is I've now got a big cell, right, and within that I've got the nucleus, and in there the amount of DNA has doubled. So what's the first thing that happens is the chromosomes line up, because that's what they do. And eventually at the end of it, we've got 2 nuclei. Okay, so you could say they line up, they divide, they're producing 2 daughter cells or nuclei. Stage 3. Stage 3, there, all right? Now, I know that is called cytokinesis. Again, you're not going to get the mark for that, okay, but there is absolutely no harm in writing it down just in case it's in the mark scheme. But this does kind of explain it a little bit. It's cell move, cytokinesis. So with that one, what you actually see is you will see divide into 2 cells. And what are they? They are identical. Question 2.6. Cell division is important in the growth of multicellular organisms. Growth. Okay, so growth, that's kind of what we're thinking about here. Figure 5 shows the mean height of boys and of girls from birth to 18. Right, let's have a look at our graph. So it starts at 0, so that's how long all the babies are, and it ends at 18, which is how long all the 18-year-olds are. Mean height. Okay, now what I can do there is I can see I've got dots for boys. There's the boys, do them in red, right, and girls, a straight line. So if I'm looking at that there, right, what I'm looking at is I'm looking at the dots and the lines look pretty much exactly the same until they get to about that point there. I'll do a different colour. Until they get to about that point there. And then there's kind of a little bit of a gap. Then I can see that what they do is they cross over there. Maybe that's going to be important. I can also see that the girls goes a lot steeper there. Maybe that's important. Then I can also see that the girls flatten out there. Maybe that's important. The boys, let's get the red out, cross over there. Maybe that's important. And the boys stop there. All right? So there are lots and lots of little different places on there where there's kind of little changes on the graph that might make you think that there's going to be something asked about. Right? Now, what I've done on this question here is I've put in the part of the mark scheme. And it says here, level 1, which is 1 to 3 marks, relevant features are identified and differences noted. So what that's going to do is it's going to be boys are taller than girls. Girls are taller than boys at the age of 12, whatever it may well be. Level 2 is when you're talking scientifically in the ways they are similar and different is made clear, right? So what you're doing here is you're looking at similar and different. And the magnitude means how much, right? So magnitude means a number.